Karibuni tena watazamaji wote wa Dr. Mlewe Lectures. Na leo hii tunaenda kuendelea na topic yetu ya biochemistry, na leo tunaenda kudiscuss kuhusu enzymes. Kwa hiyo I hope every one of you will enjoy this part of enzymes. Enzyme ni moja kati ya category ya protein which is very important in the body. Kumbuka kwenye vipindi vilivyopita tulivyokuwa tunasoma protein, tulisema kwamba we have different categories of proteins which perform different functions in the body. Lakini tukiwazungumzia enzyme, hawa ni protein ambao wako in almost every part of the body and they perform different functions in the body. Unaona? Kwa hiyo enzymes, they are very important for you to understand, and this is one among the very interesting part of topic of biochemistry. Unaona? Ukielewa enzyme, unaweza ukafurahi sana na ukapenda na hatimaye hata ukatamani sana uje usome field fulani huko mbele. Let's say usome Doctor of Medicine au usome Pharmacy. It's because only of the understanding of the enzymes. Now, enzyme can be defined as the biological catalyst which speeds up the rate of biological reactions in organism. Hawa ni biological catalyst, ni catalyst ambazo zinaongeza rate ya biological reactions. Tunafahamu catalyst. Catalyst ni kitu gani? Yani katika chemistry, we are defining a catalyst is the substance which alter the rate of chemical reaction but itself remain unchanged at the end of the reaction. Kwa hiyo a catalyst is the substance which can alter the rate of chemical reaction but itself it remain unchanged at the end of that chemical reaction. Unaona bwana? Kwa hiyo enzyme pia zinafanya hivyo. Unaona? They are catalytic proteins, wana catalyze the rate of chemical reaction in the body. Chemical reactions in the body zinaenda faster, na zikishaenda faster enzymes zinakuwa zina uwezo wa kukatalyze tena reaction nyingine. So they actually don't involve or they don't participate as the reactant, but they participate as the catalyst. Kama una concept ya catalyst kutoka kwenye chemical kinetics, it's good. But for some of teachers who wanafundisha chemical kinetics from 6 kwenye chemistry. But don't worry, might be you have the concept of catalyst from your O-level chemistry. Tulivyokuwa tunasoma mambo ya preparation of oxygen, tulisoma manganese 4 dioxide kama catalyst katika preparation of oxygen from decomposition of potassium chlorate or from the decomposition of hydrogen peroxide. In both these two cases, the manganese 4 dioxide were the catalyst. Now that catalyst which speed up the rate of chemical reaction ndio ambayo pia tunaizungumzia in this case. So enzymes are biological catalysts because they are proteins molecule made up of made by living cells. So the enzyme, they are biological catalysts because they are protein molecule made by living cells. Actually, the enzymes, they are within the cell. So hapa itakiwa ni because they are protein molecule, they are biological catalysts because they are protein molecule making the living cell au making the living cell. Hata ukisema made by living cell, it's correct because enzymes zinatengenezwa na cell. Kwa hiyo unaweza kaongea kwa maana enzymes zinatengenezwa na cell, yani made by living cell, na kama zinatengenezwa na cell manake they are biological. Kwa sababu functional unit of a living organism is the cell. So unaweza kusema kwamba enzymes zinatengenezwa na cell, au unaweza kusema kwamba enzymes ziko ndani ya cell, yani making the living cell. Unaona? So this sentence is correct in this case. A typical human cell contains several thousand of enzymes. They are used to catalyze a vast number of chemical reactions at the temperature suitable for living organism. That's between approximately 5 to 40 degrees Celsius. Most of the enzymes, they perform well their function in this temperature, between 5 and 40 degrees Celsius. Unaona bwana? Every chemical reaction involves both the bond breaking and bond making, yani bond forming. Example, the hydrolysis of sucrose involves first the breaking the bond between glucose and fructose, and then forming new bond with the hydrogen and hydroxyl group from water. In the absence of enzyme, chemical reactions in the cell would be too slow to sustain life. So in absence of enzyme, it doesn't mean kwamba chemical reaction kwenye cell zingekuwa ziendelei. The chemical reaction in the cell would continue, but their speed would be very low. Na kwa sababu hiyo ndo maana tunakuwa tuna enzyme ambazo zinaweza zikaongeza speed of this chemical reaction, and hence zikapelekea hizi chemical reaction ziweze kuwa na matokeo mazuri. So every chemical reaction nimeshasoma hiyo. Now katika enzyme au katika enzymology tunapokuwa tunasoma enzyme, kuna kitu tunakiita substrate. Very important for you to know a substrate. Now what is the substrate? Substrate, these are chemical substances which an enzyme work on. An enzyme combines with its substrate to form a short-lived enzyme-substrate complex. This proximity or closeness of the enzyme with the substrate in the complex greatly increase the chances of reaction occurring. Substrate ni ile molecule ambayo enzyme atakuwa anafanya kazi. Kwa mfano, enzyme anayedigest protein, maana substrate yake nani? Protein. Enzyme anayecatalyze, let's say, digestion of maltose, maana substrate yake nani? Ni maltose. So every enzyme has its substrate. Kila enzyme unamjua anakuwa ana substrate yake. Unaona bwana? Kwa hiyo enzyme yoyote inakuwa na substrate, ambayo substrate ndio molecule ambayo enzyme inakuwa inafanya kazi. So enzyme ukiungana na substrate anatengeneza enzyme substrate complex. Unaona? Ambayo kwenye enzyme substrate complex reaction inaweza ikatokea. Ambayo once a reaction has occurred, the complex breaks up into product and enzyme. Ile complex inavunjika tena kwenda kwenye product na enzyme. The enzyme remain unchanged at the end of the chemical reaction and is able to interact again with more substrate. Unaona? Kwa hiyo unaona substrate enzyme, tunaenda kutengeneza enzyme substrate. After that enzyme substrate complex, and then that form an equilibrium with the enzyme product complex, and then it forms enzyme and product. So now tumesha convert from substrate kwenda kwenye product, but an enzyme inakuwa imebaki pale ambayo tunaweza tukaitumia kama kawaida. If E represent enzyme, S represent substrate, and P represent product, then E plus S will form equilibrium with ES, which is enzyme substrate complex, and that will form equilibrium with EP, that is enzyme product complex, and that also will form equilibrium with E plus P. Unaona bwana? Now that was an introduction to enzymology. Na kama nilivyoongea kwenye vipindi vya nyuma kwamba most of drugs they perform function by binding the enzymes. Most of poisons they perform their function by binding with enzymes. So in the part of enzymology, you will understand a lot of very interesting phenomena in life which you could never know in any other topic of biology. Kwa hiyo tuone properties za enzyme. Kwanza, all enzymes they are globular protein, so they are protein. They are protein in nature, or they are globular protein. Mbili, being protein, they are coded for by DNA. Their synthesis is controlled by DNA. Tatu, they are catalysts, or they are biological catalysts. Nne, their presence does not alter the nature or properties of the end product of the reaction. Kwa hiyo uwepo wa enzyme haubadilishi end product. Uwepo wa enzyme unaongeza speed of a chemical reaction to occur. So the enzyme does not alter the rate of chemical reaction, but increase the speed of a chemical reaction to occur. Number 5, they are very efficient. Thus, a very small amount of catalyst brings about the change of large amount of substrate. Very efficient. Number 6, they are highly specific. Highly specific. That is, an enzyme will generally catalyze only a single reaction. For example, catalase will only catalyze, catalase will only catalyze the decomposition of hydrogen peroxide to water and oxygen. So almost every reaction in the body, it has its own enzyme. Ndio maana tuna enzyme nyingi sana mwilini. We can't mention them all. Number 7, the catalyzed reaction. The catalyzed reaction is reversible. Thus, products can reform the substrate. Enzyme wanakuwa wana catalyze reaction ambazo ni reversible. Number 8, the activity is affected by pH, temperature, substrate concentration and enzyme concentration. Uwezo wa enzyme kufanya kazi unaathiriwa na vitu vinne: pH, temperature, substrate concentration na enzyme concentration. Hivi ni vitu ambavyo vinaathiri uwezo wa enzyme kufanya kazi. Number 9, enzymes lower the activation energy. Hapa sio lower. Enzymes lower the activation energy of the reactions they catalyze. They lower the activation energy. We shall study this later in the mechanism of functioning of enzyme. Number 10, enzyme possesses active sites where the reaction takes place. These sites have specific shape. They possess active site where the reaction takes place. Now, I hope you have understood regarding the introduction and regarding the properties of enzymes. Let's move to the concept of activation energy, and let's see what do we mean by the term activation energy and how does the enzyme tend to fasten the rate of chemical reaction. Kwa hiyo tuone activation energy ni kitu gani na ni kwa namna gani enzymes zinaongeza speed ya reaction. The initial investment of energy for starting a reaction, yani the energy required to break bonds in the reactant molecule, is known as activation energy or the free energy of activation. Activation energy or the free energy of activation. Hii ni energy ambayo inahitajika kwa reactant ili kuweza kuanza reaction. Manake energy hii ni ile energy ambayo inatumika ku break bond katika reactant. Kama unakumbuka hapo mwanzoni tumesema every chemical reaction involves bond breaking and bond making. Every chemical reaction. That means this activation energy is the one which is required in bond breaking. Might be you don't know this fact. Iko hivi, bond breaking huwa ina involve absorption of energy. It is endothermic process. Bond breaking is endothermic process which involves absorption of energy. While bond making, it is exothermic process which release energy to the surrounding. Kwa hiyo, kuvunja bond tuna gain energy. Kutengeneza bond tuna release energy. Kwa tunavyokuwa tunavunja bondi kwenye substrate, lazima to gain energy. Unaona? Ile energy inayohitajika kuvunja bondi kwenye substrate inaitwa activation energy. So activation energy ni ile energy inayohitajika kuvunja bondi kwenye nini? Kwenye reactant. Au muda mwingine inaitwa free energy of activation. Maana yake baada ya zile reactant kuvunja bondi, kuna bondi mpya itatengenezwa katika product. Kama ile bondi itakuwa na bondi energy kubwa kuliko bondi ya nani? Ya reactant. Maana yake the overall reaction will be exothermic. Lakini kama ile bondi itakuwa na energy ndogo kuliko bondi ya reactant, maana yake the overall reaction will be endothermic. Hasa hivyo vitu vingine ni vya thermochemistry ambavyo ni out of scope of our topic of biochemistry. Lakini nilichokuwa najaribu kuelezea hapa nini? The concept of activation energy. Unaona, the concept of activation energy. Hopefully kwa watu ambao mnasoma chemistry tutakuwa naelewana sana tukizungumza activation energy. You have read this in your chemical kinetics, and the part of enzyme relates much to chemical kinetics and the speeds of chemical reactions, because enzymes we are studying about catalysts and how these biological catalysts they speed up the rate of biological reactions in the body. Unaona? So in this example here we have the graph of exothermic reaction. As you can see, this is the graph of exothermic reaction. This reaction tend to release heat to the surrounding. So enzyme, by functioning as catalyst, serves to reduce the activation energy required for a chemical reaction to take place. So enzyme, they reduce the energy required to break bond in reactant. In other words, these enzyme probably they can be weakened. Ni kama vile zinakuwa zina weaken, zinapunguza nguvu ya bond zilizoko kwa nani, kwa reactant, ili kwamba zivunjike kirahisi. Unaona, so for example this is the graph which show potential energy and progress of the reaction. You can observe the reactant, they start from here, substrate. In this case reactant yuko kwenye biology ni substrate, so substrate, then substrate enzyme complex, anabolic. This is the activation energy without enzyme, but in the presence of enzyme, unaona, in the presence of enzyme things are changing. The activation energy is lowered, in the presence of enzyme. So umeweza kuona difference kati ya hizi activation energy mbili, in absence and the presence of an enzyme, you see. So the change in the free energy of the reaction remain the same, but what is changed here is the activation energy required for the reaction to start. Now let's see the explanation of the graph. Before the chemical reaction can occur, molecule or substrate and enzyme must collide each other with sufficient energy, which is activation energy. Must collide each other. This is in accordance with the collision theory of chemical kinetics. If you remember the collision theory of chemical kinetics, inasema for any molecules or for two molecules to react, they must collide each other. So they must collide with sufficient energy, which is activation energy, which is usually provided in the form of heat that the reactant molecule absorbs from surroundings. If the reaction is exogonic or exothermic, activation energy will be repaid with dividends. Yani manake activation energy italipwa na baki, will be repaid with dividends. As the formation of new bond release more energy than it was invested in the breaking of old bond. Kwa hiyo in case kama reaction is exothermic. But the reaction is not always exothermic. Sometimes the reaction can be endothermic. So if the reaction is endothermic, that means it absorbs much energy, and then in forming bonds it releases few amount of energy. Na inapokuwa na release few amount of energy, that means hii reaction inakuwa endothermic. Now let's see, we have the concept of what we call the turn of a number. This is very short concept which is sometimes asked in the exam. What is the turn of a number? The turn of a number is the number of substrate molecule which one molecule of enzyme turn into product per minute. Is the number of substrate molecule which one molecule of enzyme turn into product per minute. In short, turn of a number ni ile speed ya enzyme kufanya kazi. Speed. Turn of a number inapima speed ya enzyme kufanya kazi. Enzyme ambayo inakuwa na turn of a number kubwa, maana yake ina speed kubwa. Enzyme yenye turn of a number ndogo ina speed ndogo. Unaona bwana? The fastest known enzyme is catalase enzyme, present in all aerobic tissues. Hii ndio enzyme ambayo iko very fast inajulikana. These are the highest turn of a number of about many million of substrate molecule per minute. It converts many million of substrate molecule per minute. Actually, even the rate of function of the enzyme depends on the nature of the substrate. For example, in this case, catalysts tend to catalyze the breaking of hydrogen peroxide into water and oxygen. But actually, hydrogen peroxide is very small molecule, cannot be compared to other organic molecules. This is very small inorganic molecule, and that's why catalase enzyme tend to catalyze this reaction very fast. So this is speed of the decomposition of hydrogen peroxide to water and oxygen. And if you remember kwenye cytology tulisema kwamba hydrogen peroxide ni toxic to the body. So that's why when it's produced, it's quickly broken down, or it's quickly decomposed by the catalase enzyme, and that's advantageous, manake inasababisha huyu mtu asipate madhara. Now from there, I hope you have understood the concept of turn of a number. Let's continue with the anabolism and the catabolism. Anabolism and the catabolism. Generally, the reactions occurring in the body, we can categorize them into two major categories, the anabolism and the catabolism. And generally, all reactions which are taking place in the cell, they are known as metabolism. Metabolism. So metabolism is the total sum of all chemical reactions going on in cells. All chemical reactions which are going on in cell, they are called metabolism. Metabolism, yani chemical reactions taking place in the cell, can be divided into two types, which are anabolism and catabolism. Now, it's very interesting if you have understood regarding the definition of metabolism, that it is the total of all chemical reactions taking place within the cell. And hopefully you have already understood the concept of cytology and all the organelles we discussed in cytology. I hope you know what are the rules performed by different organelles within the cell. And these rules which are performed by different organelles within the cell, they include the chemical reactions catalyzing these organelles. And now we are dividing those chemical reactions in two categories, anabolism and catabolism. Anabolism or anabolic reactions, you can whether call them anabolism or anabolic reactions. They are chemical reactions which involve the synthesis of molecule and usually require energy. They involve synthesis, zinahusisha kutengeneza. Kitu cha muhimu sana hicho. Zinahusisha nini? Kutengeneza. And usually require energy. Kwa kawaida zinatumia ATP au zinatumia nguvu, kwa sababu tukisema energy hapa tunaongelea ATP, sio energy nyingine. So they often involve condensation. Often, mara nyingi huwa zinakuwa zina involve condensation. Tunapoteza maji. You can observe this reaction, simple compounds such as carbon dioxide, urea, ammonia, or lactic acid, or ethanol. They can be reduced by the reducing ADP, and they use reduce the ADP, the hydrogen carrier, and also they use ATP, unaona bwana. And then they produce complex nutrients such as carbohydrate, protein, and lipid. The best example of this can be observed in nutrition, when you are producing carbohydrate from carbon dioxide. What happens, that is anabolic reaction. It consumes ATP, it consumes the reducing power of ADP to convert carbon dioxide into carbohydrate. And it takes place in several stages. That is another law of anabolic reaction. It involves the synthesis, yani kutengeneza kitu fulani. Then we have catabolic reaction. If you have understood well regarding the anabolic, actually utakuwa unajua tu catabolic ni kinyume chake. So catabolic reactions or catabolism are chemical reactions which involve the breaking down of molecule and usually release energy. They often involve hydrolysis and oxidation, unaona. So complex nutrient, carbohydrate, protein. Na mfano mzuri wa catabolic reaction ni respiration inavyotokea. Tunavunja glucose, tunaenda kuipeleka kwenye carbon dioxide in the water. That is the very best example of catabolic reaction. Unaona bwana. So we have complex nutrient kwenda kutengeneza kwenye simple compounds such as carbon dioxide, urea, ammonia, etc. All these reactions are catalyzed by enzyme. These two activities takes place in different parts of the cell. As we discussed, for example, photosynthesis, which is anabolic reaction, takes place in chloroplast, while respiration, which is catabolic reaction, takes place in mitochondria. So that's why the cell is compartmentalized. It is divided into compartments. We discussed regarding the cell compartmentalization. The cell is divided into the compartment, and these compartments, they perform different roles, or they perform various categories. But the major aim of all of these various activities is to enable or to ensure that an organism can survive. Now, Commonly, a number of enzymes are used in a sequence to convert one substrate into one or several products via a series of intermediate compound. Kwa mara nyingi huwa enzymes zinatumika nyingi. For example, ni glycolysis zinatumika 10, Krebs cycle zinatumika enzyme 7, 8 hivi 9, unaona. So always they are used in series or zinakuwa ziko nyingi. So the chain of reaction is referred to as metabolic pathway. Zile reaction nyingi chain zinaitwa metabolic pathway. Kwa mfano glycolysis ni metabolic pathway, Krebs cycle ni metabolic pathway, pentose phosphate ni metabolic pathway, halafu gluconeogenesis ni metabolic pathway, na nyinginezo nyingi tu. Metabolic pathways ziko nyingi sana kwenye biology. Many such pathways are going on at the same time in the cell, such as glycolysis, which is controlled by 10 enzymes. Each enzyme control only a single reaction. Now let's discuss some few differences between anabolic and catabolic reactions. You can be asked these in the exam. Some few differences. Anabolic or anabolism, this is a metabolic process that combine together small molecule to form large. These that break down large molecule into small. Require energy to form ATP. Release energy in the form of ATP. So these require energy, these release energy. Nimwambia mfano mzuri wa anabolism ni photosynthesis na mfano mzuri wa catabolism ni respiration. Occur when a cell is in the energy rich condition such as ATP NAD. Unaona, ATP NAD, cell is in rich condition of energy. Occur when the cell is in energy deficient conditions such as ATP and NAD. This kind of reaction involves hormones like estrogen, testosterone, insulin, the growth hormone. This kind of reaction involves hormones like adrenaline, cortisol, glucagon, and cytokinase. Example, the synthesis of protein from condensation of ammonia, amino acids. Wakati huku ni example is the breakdown of glucose into carbon dioxide in the water. You can observe this diagram which can show you the difference between the catabolism and anabolism and the way this ATP is recycled. From there, actually I've made this session to be very long and now I'm tired, so nitaendelea wakati mwingine. If now I continue, I'll be explaining very few explanations. So it's better I end up this session here and then another time I will continue with a lot of explanation which will make enzymology and biochemistry and biology at all the very enjoyable subject. Thank you, and let you wish you nice studies.